the first of the series. Every Wednesday of the month, we will have these seminars by ISR faculty members. And my very good colleague, Avis Cohen, I'm grateful to her for being our first speaker today. So I'm just going to tell you a little bit about Avis. Avis received her PhD from Cornell in 1977. She held postdoctoral position at the Karolinska Institute in Sweden and Washington University in St. Louis before returning to Cornell again, where she had her own laboratory studying the organization of Lamprey signal cord. And she worked on collaborative work with mathematicians, which still continues today on some of the theoretical and computational work that she has done in nonlinear oscillators. Avis joined University of Maryland, Department of Biology, well at the time was zoology in 1990, and I asked her today, she joined ISR, I believe it was in mid-90s, so she's one of our old-timers here in the Institute. She established and directed a program that is now internationally known in neuroscience and cognitive science, an interdisciplinary graduate program that combines training in cognitive and computational methods with traditional neuroscience, making this really one of the such very few programs in the country. She has collaborative work with colleagues at Johns Hopkins, and of course with a number of faculty members here in ISR, different departments in the College of Engineering. And her work is just truly exciting and is supported by National Institute of Health and National Science Foundation. And her postdoctoral associate today is joining Avis, Eric Tytell, who's giving part of the talk with Avis, and we're just delighted to listen to them. I want to encourage everyone to ask questions and make this an interactive seminar so you can get the most out of them. If you don't know much about this area, hopefully you will learn some today. And this is the trend we're going to follow. And so with no further ado, I let Avis start.
Thank you very much, Reza. It was really an exciting idea when Reza introduced this. Should I have a microphone anytime?
If you want to, yes. And by the way, this is getting taped, so the tape will be provided afterwards for those who can see. Maybe turn on the front light.
Are you getting the tape on the getting the tape? Yeah, I was very excited when Reza indicated the mic is on, but it's apparently not.
Not working. Okay, so this is one of those field things we have.
Okay, how's it now? Can you hear me now?
Yeah. Is it on?
It's on. Green light.
A little closer to your message.
Oh, it's like very good.
Very good. Okay, how's that? Okay, so just remember. And when Reza mentioned this colloquium series and that I would be the lead-on speaker, it was very exciting to me. Because I have great admiration for the company ISR, and although I don't have that much, actually I'm trying to find an optimal position for this. How's that? Because it keeps popping up.
Yeah, maybe you can just not use it.
Well, the problem is that then they won't be able to. How's this? Can you hear me with this? No, I'm going to talk loud. How's that?
Good.
Is this any better?
You can hear it.
And I'll just keep it at a proper distance. Can you hear me now?
Okay. When he mentioned it, I was very excited. Partly because I have so much, I really enjoy the people at ISR and have welcomed the opportunity to be a member of ISR for many, many years now. Because in biology, actually not that many people from biology here. How many people here are from biology? Oh, good. Okay. We have a few outliers. The problem with biologists is that they don't tend to view biological systems as systems. They think of them as pieces that one studies. And the kind of work that I've always done is very systems oriented. And in particular, this last project that I have is one to me which is very exciting and which is a grand culmination almost of my work. It is an integrated study of the neuromechanical system, the Lamprey, which is going from neurons to neuroses. And it's a collaborative project funded by the NIH, read by the NSF, because the NIH would not read these things. Philip Holmes is a mathematician and many Beck Smith, who's a chemical engineer at Princeton, Thelma Williams, who's a biologist, but is very much attuned to all the other stuff. And Lisa Fauci, who is a fluid dynamicist. So what we have here is a group that we put together, which is capable of integrating across levels of analysis, which are really not levels that the average biologist can cross. I don't know many biologists aside from Eric who knows fluid dynamics. He will be giving the second part of the talk because he, in fact, does the fluid dynamics. But he came to work with me because he didn't know physiology. And he has learned physiology and is now one of the few who knows both physiology and fluid dynamics. But the numbers are small. So I think that this kind of collaborative work, especially with this number of major PIs, is not something that you see often. You know, these are all really very senior people, every one of them. And what we've been able to accomplish, I think, and the grant is now coming to an end, what we've been able to accomplish is a remarkable amount of work. And I think it's only through the kind of effort of this kind of group that this is going to be possible. And I'm hoping that we can, with the NSF, take it to the next level. So what I'm going to talk about is environmental dynamics, which is the fluid dynamics, the sensory systems, the musculature of the animal, and the neural circuitry of the animal. We also have 10 people here. He's done mathematics on the neural circuitry, and it's been a colleague of mine for many years, is going to be working this work with us the whole time. So let me begin. The qualities of locomotion that we're all familiar with is periodic. The fact that I'm working back in the fourth year is not an accident. I don't have to think about it, which is fortunate. My spinal cord is capable of taking care of it without my cerebral influence. And please, all of you who don't know the biological terminology, because there will be many of you, so anybody who has a question, feel free, because if you have it, somebody else will also have it. So don't feel that it's just you. It's a stupid question. No stupid questions here. So please feel free to ask questions. So locomotion is periodic, and it moves the organism through some medium. It's either the air or it's water, which is the case of this critter. And it could be over the ground, which is where we do it mostly, or it could just be, flying in air, which is not locomotion. And it must be adaptive. That is to say that it must keep the animal well suited to what it's doing. It has to be able to adapt to the environment as it goes on the fly. This is critical. So there has to be sensory feedback. There has to be all of this integration of information in order for it to work properly. And one of the things which is often lost by biologists, is that the mechanical properties of the organism have to be used efficiently. And this is often what's forgotten by biologists, because most of the biologists, either they know the mechanics and they don't know the neuroscience, or they know the neuroscience and they just sort of ignore the mechanics. But I think that the time has now come to put the system back together, and that's what I have been able to do with the help of my colleagues. Historically, there were two sides to the studies of locomotion. In the early 1990s and onwards, there was discussion of a neuron pattern generator, and it dominated the discussion. There was in the Russians in the 60s, and Mrs. Bernstein, who some of you may be aware of, and then Wilmer in the 80s, and he was my postdoctoral lecturer. The sensory feedback was dominant in his discussions. In the 50s and onward, with Keir Pearson, and these two were two sides of the discussion, which I always thought was kind of weird, because of course you need a hope. I couldn't understand why you would want to cleave them apart, but this was how it was discussed. It was one of the, I think there's certainly at least one seat here, and please, so these two sides argued a lot in the 80s. But then, in the 90s and since then, we've had a new generation of young people who are integrated. They want to arrive at new synthesis. So you've got, these are both colleagues, by the way, who are in Europe. Ansgar Buschkis is in German, and Albert Eastbert is from Switzerland. And both of them are looking at this kind of new synthesis. But it's difficult. It's difficult for the reasons that I have said. So on top of that, you have control issues, which add an interesting take. And Bill Levine and I just had an interesting conversation on this just the other day, because in fact he has a very interesting take on the control issues that may start to come up now. That's right. I think it's very interesting. The organism that we have looked at is the relatively primitive vertebrate, the Lamprey. It's what we call in biology the basal vertebrate. That is, if you look at the vertebrate tree, it's at the bottom of the tree. So it's at the base of the tree. And we don't talk about it as primitive, really. We talk about it as basal. It's, that's just sort of a biological thing. It has a relatively simple nervous system, which is very nice for those of us who study the system. Spinal cord, for example, is transparent, and it's easy to study because it doesn't have any circulatory system in it. There's no blood vessels that go into the system, which actually is leading me in another direction with physicists with whom I'm looking at putting it in a beam to look at the blood-brain barrier. Because in fact, it's getting the whole notion of a blood-brain barrier because it doesn't have a blood system, so somehow it's not really. But anyway, that's an aside, and in 5 more years I'll get back to that story. However, it is a good vertebrate. It's a real, honest-to-God vertebrate. At the base of the vertebrate tree, there are actually 2 critters. There's the lamprey and the hagfish. And the hagfish is what it sounds like. It looks like a hag. I hate to use that term because it's a derogatory term for a woman. But this is the ugliest creature ever found. But it is not as good a vertebrate as the lamprey, thank God, because it's really a very unpleasant creature. So in any case, that's down there at the bottom. But the lamprey, I used to have one of my other postdoc mentors, Carl O'Reilly, who is now off music somewhere on the West Coast of Oregon. He used to say that the distance, evolutionary distance, from lamprey to human was smaller than the evolutionary distance from lamprey to Amphioxus, which is just below the tree, just more basal. So in fact, It really is a very good vertebrate, and it has all the features of good vertebrates. So, in fact, if you look at what's going on in the vertebrate, we can assume to first order, and I emphasize this is really to first order, that much of what we find will hold in other vertebrates like mammals, even humans. And that's a stretch, and you have to prove it. But at least it is suggested by this one. Okay? So that's what we're doing. Now, the behavior is this. This is swimming, and this is a little guy. It's actually an aniseed, which is another term for the larva. It swims pretty much like the adult. You'll see an adult later. But this is wonderful. There's no big difference between, again, to first order. This is even to capital between the aniseed and the adult.
So what's their lifetime? How long do they live?
They actually live quite a long time, which makes them very hard to raise. You can't raise them. The aniseed lives for 3 to 5 years or longer, depending on the temperature of the water it's in. Then it metamorphoses, and the metamorphosis takes 6 months, which is really a pain in the butt, because they don't always make it in the lab, because they don't want to live in the lab, they would rather live in nature. So it's not easy to take the transformation. And then they go into the adult phase for 1 to 2 years, and the major change between the larva and the adult is a change in eating. As the larva, it eats its filter fish that lives in the mud with its head just sticking up out of the mud, and filter foods. The adult is has a sucker mouth and is a predator of fish like trout, salmon. So it's very unpopular with fish. So we have to, when we catch them, which I used to do with Ithaca, I had to promise the fisherman that I would torture them to death. For them to give them to me. There was no love lost between my providers and the animals. On the other hand, it is a vertebrate, so I must provide the NIH with all of the forms. So what we're going to do is this is what we do. That's fine. It's important. So that's our letter. Now, we start with the neuronal network. And, but which one? Because in the Lamprey, there is, this is sort of a, shall we say, a truncated or simplified version. And this is a less simplified version. This is from Griller and his colleagues. And what he has done is take a very simplified version of what he has found in this. So this is what this is from the brain. Now, David Parker at Cambridge has, in fact, found that there are many, many more cells than really wants to acknowledge in this circuitry, making the system much more difficult to study than really wants it to be. So the acknowledged system is this one, but the real system is this one. So,
Neurons?
yeah, those are neurons. And what David has done is he does very detailed studies of poking one of these and then one of these and showing how they're connected to each other. And he's also done these little guys, how they're connected to these, and how they're connected across the cord, and so on. And it's very painstaking, very difficult work. And nobody really wants to do it, except David is very patient. That's right, and they're all up. That's right, and that's the other thing that Emily wants to sort of sweep up. So this is the acknowledged, or the accepted.
Different colors or what's important?
The different classes of neurons. So, in fact, David emphasized that they are classes and not, that there's more than one class in each one of those.
which is something that again. But there's 1,000 neurons in a segment. You're not going to have, and only 100 of those are the motor neurons that go out to the muscle.
Is there a map from one to the other, or?
Well, in here, if you look, there's the L, the I, and the E. L is this guy, the E is this guy, and the I is this one. And that's the map. But in fact, what David has found is that there are many more types than just one. And that's what we would like to know. And it's unfortunate because this box is the 4 boxes. Oh, these are. This is a different. It's a different map or a simplified version of this. I haven't looked at this for a while, but I think it probably comes down to a simplified version of this. He had several versions, several different varieties of carcinogens, and I just. So I don't remember. Yeah, but whether the A and the B are different versions, it was a while ago. So, okay, now, so that's the different models. Now, working with mathematicians, we have no such problems. We just talk about phase models. That's all. Just throw it all the details. We're going to talk about a phase model. No explicit biological details. Each segment is represented by a variable. This is what we've done since the 1980s. We started working with this. Now, in the first order phase models, what you get is a good approximation of the system. If the system is connected, so the thing that I haven't said is that you have 100 segments in the animal. And the segments, in order to give you a traveling wave of the activity down the cord, you have to connect those segments. And they're connected. And I know they're connected both asynchronously and desynchronously. It's not just one wave coupling, which is what people like. No, it's like. It's actually two wave coupling. And I believe that, and I was in this last. Unfortunately, that makes it very difficult to think about because this one is sending a signal, this one is also sending a signal. So it's very complex. It cannot be thought about in a typical, well, this does that, and then that does that, and then so on. And this is what biologists would like. So mathematicians that I went to when I came back from Sweden, there was Phil Holmes and Richard Rand, and Philip had just gone to Cornell, so he was in the project. Hey, Phil. How would you like a project? So we started working together, and as we say, the rest is history. We published our first paper in 1982, and now he's been working with that. And Tim is a student at Phillips, and has been working with it ever since. So he's here if I have any questions about mathematics as well. And we know where our strengths are. Because mathematics. So, the first order phase models give a good approximation if we assume, or if we can show, that the connections between segments are weak. Now, what we have been able to show using physiology and modeling, using Tim's mathematics, maximum likelihood methods, and various other kind of methods, we have been able to show that, in fact, weak coupling is not an approximation. In fact, it is strong coupling. So what he's had to do is go to second order phase models, and we'll talk a little tiny bit about that. But they nicely indicate how the behavior changes as the coupling strength begins to increase. And this is new work of his, which is very, very useful in exotic coupling. Now we can use these phase models, or we can also go to simplified connection spots. That is, you take a little ball of stick and imagine, and you represent that neuron is just a little stick of connection. The classic neurons
by a single variable. And here, Thelma Williams is one of our collaborators. She has a model which uses 6 neurons in this type of process. And that her model was published in Science in
92.
Here's the question. What is weak coupling and strong coupling?
Yeah, so the difference, one of the main differences is if you think about an input, say for example, somebody were to I go, that's strong coupling, that input. Right to that. Or if you're able to disconnect your arms and your legs, which you can, that's very, but for example, I can rub your arm. So I can disconnect. So that's a weak coupling. Strong coupling is if I have a choice. And it always
Strong and weak don't refer to how one segment connects to the adjacent segment.
That would, because there, my arms and my legs are like the different segments. The sensory input was the first one, and that was sensory input to the spinal column. But the arms and the legs, that's the segments. That's these segments to these segments. And the animal has these 100 segments, we have this traveling wave of activity. And in order to keep that traveling wave of activity, which on the slides from up, in here, you can see the traveling wave of activity part. But actually, let me just go back to the animal. And you'll see. You see there's a traveling wave of activity that runs down the animal body. That's the traveling wave, which the connectivity responsible for. And that is, it's strong coupling in order to provide that. This animal doesn't have much flexibility in its coupling. More derived animals, which is the proper way of talking about us, is we have a lot of flexibility in our coupling. So we can disconnect it, we can reconnect it. But it will with the brain. And that's it. Change the coupling for us. Right? Okay. So that's the connections models. Now there's biophysical models where each neuron is represented by multiple variables and not the top ones. That's the yes.
Are we restricted? So say models of CPG, these are only for Lamprey, or are these in general CPG models?
The model for the CPG of the Lamprey is, well, we know, for example, that the tadpole is similar. And we don't know, the problem is that it's very difficult to get the structure of the spinal cord down to the level of neurons in anybody else. Because in the Lamprey, there's only 1,000 neurons per second. But think of it. In somebody like a cat, or even a rat, now there are people who are studying these, and they're using genetic methods to knock out the classes of cells. That will start to disentangle the spinal cord to some extent, but it's going to be very hard. So we really don't have, all we know is that it's coupled, that each segment is capable of producing an oscillation, each hemisegment, and that there's a coupling across the hemisegments that we know. That's, this animal, and it's all animals, very strong, but it's variable. In this animal, it's probably also variable, not as variable as us, for example. So, yeah, but that's actually a very good question, and we really don't have a clue because it's just such an impossibility to study the details. Yes.
In tinier creatures, the nematode swims something the same way. Is there half the nematode of the lamprey?
The thing with the nematode actually is the physiology is so different that it's very difficult to generalize. I have not sat down, and there is a guy over at NIH that I know quite well who studies the nematode, just looking at the connectivity. But the physiology is really just totally different. So I don't know anything about it. How it's
CPGs are in abstraction.
absolutely. That actually has been a hotly debated topic. There's no question. The question is, how do we know there is a CPG? So, the definition of CPG is part of the argument. The argument is that there exists a network. I will not argue that it's a fixed network, because in fact I would argue that it is not. It is in the spinal cord, and is the organizational structure, if you will, of the spine, all the way up to vertebrates, to mammals. And this has been, there's a guy at NIH, Michael Donovan, and he has shown that there is, even in chick embryos, that there is a structure which is doing what it needs to do in order to be a CPG in all the segments of the spine. You can activate it through various methods, and then you record the activities in muscle. And that's what he's doing. And what you see is alternation, here you see sitting here, that's really all you can see. Beyond that, it's very hard to go through. They've used the chick, they've used the chick in order to show that the chick embryo has fractionated to give limb movements and muscle movements that are appropriate and at a time which is remarkably early. So even that much is in there at birth and not even prior to birth. So when a baby is first born, I remember when our first son was born, I was so excited. So where was he belong? The fact is that that's actually true. Because what happens is that as they develop, the brain comes down and turns it off because it's not strong enough to maintain it's dead bones. So, no. But in any case, it's there at birth. So I don't know what structure it takes. I don't know, we know it's distributed throughout the spinal cord. Is it every segment of the spinal cord? I would claim it is. And I do it.
This picture here in much simpler creatures like sciatic animal, like a lobster, or locust.
Yes, it is. And there, but the problem is that we don't know if it's the same system. Because there's such a distance, evolutionary distance, that I actually wrote a paper in '87 on comparing the stomatogastric ganglion of a cat and lamprey. Because it seemed to me that what you can look for, that you can find, are organizational principles that hold your nervous systems. Are they done the same way in all these animals? I have no clue.
For mathematicians,
Yeah.
the existence of one CPG would be an existence.
I agree. But
Even within a-
yeah, I agree. But the problem is that how they are built is going to be different as you go across the animals. They all have to oscillate. Every animal in the animal kingdom has to have some form of oscillation. And the methods for producing those oscillations are all well, they're all different to some measure. I can prove that. But then how they're used, how they're controlled, and all this stuff is, that I think we know.
How about signal transmission? Fixed has
Signal transmission,
mechanical.
yeah, that is fixed.
Obviously.
Always fixed. Because that depends upon the CPT questions addressed. Numbers. Okay. How do we do the intersection coupling using all the details, if not which ones? So there was some of the simple phase oscillator, and we've also started working with Hoffman, who's over at UMBC. She does a very nice system. We'll talk about it in 2 seconds or so. And the first order phase model is this much here. This was what we worked on with Phil and Richard. And that is being now added forcing term to that, which is this term. How do I get what?
How do you get the values?
You can study this.
Yeah.
You can study this as a mathematical.
Yeah, we
It's
even
not
do
data
it.
independent.
Tim, you can talk to Tim afterwards. Because it's not interpreted.
I'll bring it up later, but one of the points I'm using these very simple models is we don't know about it because of the Avis stage, but can we use the models to make general statements about how they might be structured? And I'll show one result that I think we may be able to go in and make some biological measurements to see whether the structure is there. But of course, the actual values are guesses. We're interested in the general structure.
Yeah, the thing is, again, you've got neurons that are making these connections. I can't give you the values of individual neurons. I can tell you the effective functional strength, but that's a composite. N is number of seconds. It's up to 1. Okay, it turns out that it's interesting because, in fact, one of the things that I showed many years ago is that there is a natural frequency that the different parts of the spinal cord have, and they're different. So there is a natural difference in frequencies along the spinal cord. It's not linear. There's no gradient. It's a mess. So it's a complex system.
For simplicity, we assume it's the same. In fact, we know experimentally that it's not.
But, again, you have to make some simplification go down the paper. So phase model versus full models, this is preaching required here. The stable nonlinear, for phase model, we're assuming a stable nonlinear limit cycle oscillator described as a parameter. The full model assumptions, when we do a full model, are based on the connections model from Williams, and that's this system here. And it comes out of the paper. Oh, I'm sorry. That's great. Okay, now using, now Tim has recently done stuff with second order coupling, and that's actually made it much more interesting because these are just with first order coupling. I'm not going to go through this at all, but suffice it to say, the second order coupling, you get some of the phenomena that you cannot get with first order coupling, especially because you can't get moderate coupling and maintain what we know is there with moderate coupling, but with moderate coupling in second order we can't. So it's I'm very excited. It really it's going to give us much more. This is the equations for the second order phase model. It's up here, it's there, it exists. Love opportunity. Look at it every time, like 10 minutes. Another nice thing that you have with second order coupling is that you now have these maps, these three-dimensional maps, these vector fields, where you can begin to talk about how the system changes as you move through different coupling strengths. And it's kind of cool because it means that you can look in different ways and how the system is changing. Yeah.
Second order dynamic model is second order model first order done. Here at all, not here at all.
Yes. That you're just adding a coupling term, which now goes from, go back. Yeah, it's like, so here, you have, this is nearest neighbor. I'm sorry, I should thank you for asking the question. The original models were all nearest neighbor. Okay, and that was first order coupling. So second order coupling means that you add this term here, and these additional terms will accompany which will be further segments. And that was not in the original connections, the original base model. Okay, so that's what it is. Okay, so some of the advantages of the second order coupling are that it allows you to see how the coupling strength influences the behavior of the system, because we can put in strong coupling with the second order coupling. We know that there's strong coupling. So that's pretty very nice because we couldn't do it just first order. Second order coupling is also, we derive the first and second order using averaging. And again, this is something that's in M's mathematics, right? The mathematicians have used NASA. Forcing those second order terms, forcing is like when you move it yourself. If somebody were to swing my leg, that would be forcing my leg. And that's what we're going to do with the Lamprey. So you're forcing the leg. You're forcing the spinal cord. That's what it tends to do. Errors. Okay, and then Kathleen is interested in also. She is introduced a very nice new way of doing coupling, which has this random coupling. And it's really pretty because this is probably the way the system develops in the animal. So what she does is she lets it be a randomly distributed crossroad distribution, making nearest neighbor more likely, but the length of the coupling is determined with the geometric distribution. And she uses these rules, and it's very, very good. And since it is much more like the real system would be doing in the development of the animal, it's really nice to see this. So I think that's going to be really nice. She is working on some very stable. So, all organisms must sense their environments during nutrition. Sense organisms must convey the most critical information on what's around and near them, like examples of prey, where they are, how they are moving in their environment, where they're moving in their environment, other relevant properties of the medium, whatever that medium happens to be. So, we have two sensory systems that we can talk about in Lamprey. Only one is really known to affect locomotion, and that is edge cells. These are mechanoreceptors intrinsically to spinal cord. We don't have them. We have what are called spindle organs, which are mechanoreceptors in our muscles. The lampreys don't have spindle organs, but they do have these loci, which I'll show you in a minute. But there's some possibility that snakes have them. But they really are not found in anybody else. Other possible sensory organs is the lateral line system. And this one could, in fact, really be involved, especially in the real live animal, because it effectively is measuring turbulence of the animal. So, and the animal certainly is swimming in turbulence at various times. Others, we simply don't know. Nobody wants to know. This is a measure. This is a picture. It's a beautiful image. And you actually see the little feet. This is the cell body, and then these little feet go right up to the edge of the spine. And you just there's a little feet in, and they can stretch. So they're beautiful. It's known that they can train the swimming. That is, it's known that if you take a spinal, just a plain spinal cord all by itself and wag, that swimming will become trained by that way. And since the only thing that's there is the stretch receptors, these edge cells, it's got to be the edge cells that are doing it. So we knew right off the bat that they were doing it. So Tim and I also have shown that they activate a slow excitatory activity on the spinal cord. Okay, and now I hand it over to Eric.
Okay.
And then we'll come back.
We're going to jump around a little bit here.
I understand.
So in a moment, I'm going to show you a video here of the rawest of raw data. It's actually a video of my experiment in operation. But I'd like to point out what's going on here before I started. This is a spinal cord sitting in a dish. These are electrodes that are measuring the motor output that would go to the muscle if I hadn't dissected all the muscle off. And this little device here will bend the spinal cord back and forth, stimulating those edge cells. You're going to hear through the speaker the output of one of these channels. You'll hear zzz. That's when the motor output would go to the muscles if there were any muscle left. And so you can hear the fact that it's in training. So let me play that. So that zzz, that's the motor output.
The lower frequency is the motor output.
And the wee-oo, that's the motor. And this is slowing in frequency now, if you listen carefully. So it's matching the frequency as I slow it down. And I'd like to point out, first off, that this never happens in reality. This is a completely artificial situation because normally the CPG stimulates the muscles, which bends the body. It's always entrained to itself. But we're using this artificial stimulation protocol to see how the system behaves. Oops, now I'm going to play it again. Yes?
These are bad electrodes, or?
These are suction electrodes on the ventral root. So these are multiple unit electrodes. Now, one of the things we can do with this is we can look at the range of frequencies over which the CPG will entrain. So this is an example data set here. This is frequency of bursting in yellow here. Here's the stimulus, this bending back and forth as I ramp the frequency up. You can see when the stimulus frequency is too low, it doesn't match the frequency. And then after a while, it starts to match the frequency of the stimulus for a period, and then it stops being able to match it again. So this is the entrainment range. There's an upper bound to the range and a lower bound. And if we map those out by stimulating at various positions along the spinal cord, we see a relationship that looks sort of like this, where these red downward pointing arrows represent a lower bound to entrainment, and the blue upward pointing arrows represent an upper bound. The example we looked at are these two right here, this one there. And the thing that I found was that there was a somewhat larger range on average towards the middle of the cord when I bent there than out at the edges. And I thought about this and I said, well, that seems natural. After all, if I bend at the middle, the signal has a shorter distance to travel. It makes sense. It should be stronger by the time it reaches the edge of the cord. That's probably why it's a larger range in the middle. And so together with Tim Kiemel and Kathleen Hoffman, we went in and put together one of these very simple phase models to try to explore this effect. So here again are the equations for the phase model. This is just an example of nearest neighbor coupling, where each of these dots represents in a segment, more or less, and they're coupled only to the one nearest on the chain. And then we have a forcing oscillator here, which forces one of those oscillators, provides an input to one of them. For the moment, we're only going to think about nearest neighbor. This is also work done with Joe Previn, Penn State Erie. And what he was able to put together was a few conditions for entrainment. So entrainment, if we have our forcing oscillator, so if the chain by itself wants to run at 1 hertz, that's what all those 1.0s mean. And then I have a forcing oscillator that wants to run at 1.1, that forces it at 1.1 hertz. If it's entrained, the whole chain will match that 1.1 hertz. The whole thing will be running at 1.1 hertz. If I then up the frequency, there we go, to 1.2 hertz, suppose that's outside of the range of entrainment, then there are several things that could happen. One thing that could happen is that the chain would no longer match the frequency of the oscillator. So this wouldn't be exactly 1 hertz, but it wouldn't be 1.2 either. So this is no longer entrained in the oscillator. And this is called an external loss of entrainment. So the external forcing is running at a different frequency than the chain itself. There are also 2 other mechanisms for losing entrainment, and because we're low on time, I'm not going to talk about them at all. In general, in experimental situations, we only see this external loss of entrainment. So we're going to think about external loss of entrainment here. Now, Tim went through and did some more work on this and found that when we have long-distance connections, so now we're no longer thinking about nearest neighbor connections just from one oscillator to the other, but potentially long connections along the link in the chain. In fact, all-to-all connections. So every oscillator is connected to every other oscillator. If those long connections have the following form, such that the long connections are some relationship to the nearest neighbor connections, these alpha 1 and alpha minus 1 are the connections to the nearest neighbors. So for example, the long distance connections might, the strength of those connections might decrease exponentially with length. Then this inequality that we proved for the nearest neighbor connection holds even when we have long distance connections.
Next question.
Yes?
So, in the 2 slides back, in the equation, you had for each I, you had a summation over all k of alpha sine of alpha I minus alpha k, something like that, for each I. And you had the sum, one I think is 1 4, right? So we were talking about pi dot, but you have the sum over all j of phi j minus pi. So isn't that long distance? The j,
Yeah, so this is the general model.
that is long distance, isn't it? The j.
Exactly.
Oh, it is. Okay.
So in the nearest neighbor thing, we take our alpha i's to have this form,
Oh, okay.
such that there's only an alpha 1 and alpha minus 1. And we do some work on that. And then we can extend that to long distance calculations where our alphas have terms outside of this alpha 1 and minus 1. Here. Now the trouble is, if this condition holds, then numerical simulations of the entrainment ranges have a form that looks like this. So this is the frequency difference between the forcing oscillator and the baseline frequency of the chain, the ones to run at with no forcing, as a function of where we're applying the forcing. So we have a 50 oscillator chain here. If we force at oscillator 50, we get an entrainment range of about plus or minus 10%. If we train at the other end, we get plus or minus 5%. And that's always monotonic relative to the forcing.
Is there any limit on the connection line?
Well, here it's all to all. So the connections go from every oscillator, every other oscillator. So this is a 50 oscillator chain, so there will be connections according. The trouble is that this doesn't look very much like my data. My data had this hump in the middle. And it turns out not to be a trivial operation to get a bump in the middle of this sort of relationship. What we've proposed, and this is still a conjecture, we actually haven't proven this yet, is that this is related to coupling asymmetry. And what we mean by coupling asymmetry is that if at length 1, if ascending connections are stronger than descending connections at length 1, then they're also stronger than ascending connections are also stronger than descending connections at longer lengths. So this is a uniform coupling asymmetry. If we show the ascending strengths here, they're always stronger than the descending strengths here. And numerically, at least, this gives monotonic entrainment ranges as a function of position. Now, if we take a non-uniform coupling asymmetry, such that ascending connections at short distances are weaker here than descending connections, but at long distances, now ascending connections are stronger than descending connections, then we can start to see, numerically at least, entrainment ranges that have multiple elements.
You're only considering the neuronal connections?
Yes.
But are there not physical connections to the spinal cord
Absolutely.
that are going to then be excited by the same little organs that detect pressure of the spinal cord when it's excited? So you have a whole other layer of connections that will for sure drive the entrainment.
Absolutely. Yeah, so this is artificial in a number of senses. One, there are no mechanical connections among the oscillators. Two, this is an open loop situation. In fact, the real system runs in a closed loop. You can't talk about entrainment in the actual system because it's always entrained to itself. What we're trying to do here is to use this very simple model to tell us something about the structure of the neural connections. What it seems to be indicating is that they're not in this simple, uniformly asymmetric form, that we probably have some sort of a non-uniform connection. There are lots of other things that will be going on, and as we get into our more into our fluid dynamic models, we'll have some more detail on that. Okay, so do we want to talk about model one?
Yeah, let me just.
Okay, so we had a whole other sensory system we were going to talk about. We're going to skip over that. It's the lateral line. It also seems to feedback onto the central pattern generator. We have a nice model of muscle. It was. Okay, you want to talk about making it work? Okay, I'll just take the last. Okay, so we have a model of the musculature of the lamprey that Elma Williams has been putting together. This is based on data experimental data from the lamprey. And we're starting to connect this in a closed loop fashion from our central pattern generator models through our muscle model back to feedback on the central pattern. Now finally, we have to consider fluid dynamics. This animal is swimming in water. This is the experimental work has primarily been done by Lex Spitz and his students at Princeton. And we started with some early attempts to get lampreys to swim and die. The challenge here is that lampreys really don't like to swim steadily. They have this nice sucker mouth, and if they don't like to do what you want them to do, they simply suck onto the side of the tank and you can't really get them to do anything. So we had some at least moderately interesting results, but nothing we could analyze in any great detail. You can see the flow separating as the animals.
What's the Reynolds number?
The Reynolds number, these animals swim at something on the order of 10,000. Depends, of course, on the smaller ones might be down as low as 100. The big ones could get substantially higher. But it's all high limits.
You are getting vortices.
Oh, absolutely, yes. So we decided, instead of trying to work with real lampreys, we'd build an artificial one.
And Lex's group is building it for us.
So this has been Lex's group who's built this artificial lamprey. This is kind of a monster lamprey. It's about 1 meter long, and it's not quite matching due to the limitations of their flow tunnel. It can't beat its tail quite fast enough to match the reduced frequency of the tail in an actual lamprey, but it's not too far off. So this has been primarily Megan Leftwich, which is one of Lex's students, who's been working on
It was
this.
recently bifurcated to understand the team.
So she had her baby, was it on Saturday? So this is data from immediately prior to the birth. So she has put together this robot which does a pretty good job of replicating the anguilliform swimming motion of a lamprey. She's been working on the pressure distribution along the swimming animal, and just recently she's been doing some work on the role of flexibility. So she's been attaching flexible tails to the end of this robot. These are some examples of the tails. This is the original tail, which is a very stiff square tail. She's now working with tails that are based off a CT scan of a swimming lamprey, or of a lamprey, not swimming. And she's able to vary the flexibility of the tail there. That's getting some very interesting results.
Are there muscles in the tail, or is it passive?
The tail is actuated basically out to the tip. So they're fin rays that are maybe the last millimeter or so are activated only in the base of the fin rays. But pretty much aside from that last tiny little bit where they're fin rays, it's active. Now the muscle is there's not a lot of muscle, but there isn't.
How do you quantify similarity to these swimming behavior? And
So-
also, I can actually add something to that too to help me answer if that.
Okay, so the main parameters we're looking at are Reynolds number and Screw-Hall number. Screw-Hall number is the reduced frequency of the tail beat motion relative to the forward speed. We also have the amplitude envelope, that's about right, in terms of how the amplitude flows along the body. Those are the main two.
Okay. And is the goal to model or to mimic the animal as closely as possible, or to meet some other metric?
The goal here is we want it as we want to mimic the animal as closely as possible so that we can use it as in order to make measurements that we couldn't make on the actual. So if we wanted to be, we're not looking to optimize efficiency or something along those lines. So the other route we're taking so very quickly here, the other route we're taking to look at this system is to put together a fully coupled computational model. So this is a fluid dynamics solver coupled to an elastic body. So here is a schematic of the structure or a computational Lamprey. It's two-dimensional at the moment, but we have the body here. It has kind of a backbone and the lateral edges. These lines represent springs, so they're all coupled together by normal Hookean springs. And we have the activated segments here that produce force according to a model of muscle. Then we put this in an immersed boundary framework. This is a fairly standard computational technique at this point. We have our swimming sheet in a computational domain. It applies, sorry, was there a question? It applies forces at each of the points along the body. These are Lagrangian points. We apply forces from those Lagrangian points to the fluid. We transmit that to the grid, solve the Navier-Stokes equations on that grid with that extra force, interpolate the grid velocity, the fluid velocities, back to our Lagrangian points that define our body, and update the time step.
What I was wondering about is Charlie Peskin, very famous for his study.
This is Peskin's method. You can see Peskin, Active America. So this is Lisa Fauci doing most of this work. She was a student.
Working with the postdoc, Chai Yu-Shu, to see pictures of her at the end.
Okay, so these are the equations. I'm not going to tell you about them. Conceptually, here's what we're interested in. We're interested, what our model is able to do that no other model at the moment does at this level of resolution is to consider the effects of the body stiffness and how that couples with food. So this is a picture of a Lamprey tail. Here's a block of muscle. It exerts force on the tail in some way and causes it to bend. Now, there is a restoring force here due to the elasticity of the body. There are also fluid forces that act on the tail. Now you can imagine kind of an extreme case where the fluid forces are quite large and the body is quite floppy. So this means that the fluid forces are actually relatively very important for determining the shape this body takes as the muscles deform. There's an alternate condition where the body is rather stiff, but the muscles are rather strong, such that these body forces are quite large in comparison to the fluid forces represented by this little arrow. And so the fluid forces are not terribly important. Now the extreme model of this is the prescribed kinematics model, which is what most fish swimming fluid dynamics models have been, where they prescribe the motion of the body and then solve the fluid motion on the basis of that. But the fluid forces on the body have no effect on the shape the body actually takes, because that's prescribed in the simulation. So ours is the first model that's fully coupled and allows the kinematics to emerge from balance these internal and external forces. There have been some recent models where a joint has been coupled, or a few joints have been coupled. So that a number of different groups are starting to take this approach. But to my knowledge, this is the first one where we have a completely elastic model. Kinematics really do emerge from the forces that we're prescribing, rather than. Here's just an example. This red portion is where the muscles are active. This is just to show you how the wave of activity travels in the body. We have this hill muscle model embedded in there. So in fact, we don't even prescribe muscle force, we prescribe muscle activation. And in order to get up to an appropriate Reynolds number, at the moment we're running up to about a Reynolds number of about 80,000 based on body length. We used a spatially adaptive grid solver that was initially developed by Boyce Griffith at NYU. This example here just shows the blue boxes are where the grid is increased in resolution. So it follows both the body and the vortices in order to maintain appropriate resolution.
Not as much head
It depends on a number of parameters. Well, I'll show you some examples. So here is kind of our normal case. What you'll see is, so I'll start this thing swimming. The red and blue represent vorticity in the fluid. This is the swimming speed over time. And this is the amplitude envelope of what you're watching here. So this is the maximum amplitude any given point on the body, and this is to show the body weight as it swims. Now to show you that these kinematics really are emerging from the simulation, they aren't something we pre-programmed, I'm going to change one parameter, which is viscosity. Everything else is exactly the same, and I'm going to run it a second time. This is high viscosity. What you'll see, now I've overlaid the body weight here. The red represents the high viscosity case. You'll see it's struggling a great deal more, which is not surprising. And it swims slower. But the most interesting thing to me is that if you look at this red trace here, the wavelength decreases in the high viscosity. These kinematics really do emerge from the coupling between the body and the external environment.
That's like a younger Lamprey.
That's right. Actually,
Yes.
it is.
The viscosity change is not, it was a factor of 10 change in viscosity, and for a younger Lamprey, it's not going to be quite that big. But it's maybe a factor of 2 to 5.
So the young Lampreys still use the same strategy or whatever?
Yes.
To swim?
Yeah.
They just test it.
It's probably less stiff.
We don't know how the material properties change.
Was it not, what direction is it trying to go? Does it look like it?
It swam off to the side. Yeah, so we have an interesting problem that since we don't prescribe our kinematics, we have to kind of run experiments with our simulation to find out what it does. And initial conditions matter quite a bit. So when it kicks its tail to one side, that sets up a slight deviation at the beginning of the condition. It swims off at an angle again.
The control engineer, that's just mother's milk, right? Because that says that you've got to control it or it ain't going to go where it wants to go.
Precisely.
So there's got to be another route.
And that's the final. So we have this thing so that we're only prescribing muscle activation. Well, that's the output of our CPG models. So fairly soon we're going to be putting this whole thing together and having a full loop with the sensory field. Now, to finish things off here, I just wanted to, since I introduced this with the idea of relative stiffness and body stiffness and fluid forces, we'll finish this off with an example here of 3 different cases. Here's a stiff case where the internal forces are relatively large and the fluid forces are relatively unimportant, and a floppy case where the fluid forces are relatively important. and the internal forces are less. We're going to erase them. So our stiff-bodied model is first off the mark. Our floppy-bodied model is not doing too well. And as we move on, what was interesting to see actually is that, so the stiff-body model here in light blue, in fact, they all reach roughly the same steady swimming velocity. The stiff-body model accelerates a great deal faster, but in fact, they swim at roughly the same speed. And the kinematics are pretty simple. Here's the stiff model, the floppy model, or the normal model, and the floppy model. The floppy model has a slightly shorter body weight. Now, if we come in and we look at what's going on in the steady state condition here at the end of each trace, we can estimate the work performed by the muscles. This is the total energy input for the stiff-bodied model, the normal one, and the floppy model. And we'll see that once they're up to this steady speed, which is more or less the same, in fact, the floppy model is working a lot less hard to maintain that speed. In the same way, if we look at the fluid forces, this is the total impulse over a tail beat, the stiff-bodied model in light green here is producing a lot more thrust, but also a lot more drag, whereas the floppy model is less thrust but less drag. So in fact, we're seeing for the same neural commands, it's exactly the same traveling wave, but we've changed the body properties. In fact, we get rather different performances. This stiff-bodied model is an accelerator, maybe it's like a pike, whereas this floppy model is better at efficient swimming. So I'll let Avis tie the whole thing together.
Okay, so there you have it, pretty much. So what we've talked about is taking phase models to capture the CPG, muscle output, edge cells for the sensory part, mechanics. We're taking from the robot what the movement is, and we're looking at all of that with the CFD computation fluid dynamics all together, and we up with mom. And this is a group, there's the homes. Couching, this is actually students from John Esmond's Chinese picture. We don't have this slide. Here's like Smith's, there's Williams. Who's now emeritus and doing a great time working on stuff that she's doing. Here is our whole group. At the moment, there's a couple people who have led us to China. There's Lisa, Elmo, Tim, Kathleen, Lex, Stephen, and Jeff. Megan, and this was last weekend, and she has since had a baby, so it was a very exciting meeting, just in time. She got to the hospital. So, it's been great for me, very intensive ongoing. Thank you.
Thank you, Avis and Eric, for such an excellent seminar. I think we have time for some questions.
Yeah, and people have to leave, I understand.
Yeah, you have to leave, that's
I was
okay.
wondering, in the broader picture where you look across species, do you want to use the notion of a virtual CPG? In other words, we should map to where it will work in a given case. For example, you have random connections in your Lamprey.
Yes.
So you're mapping a CPG onto one of the pre-existing structures. The CPG might be very simple in an abstract sort of way, but mapped into something which could be quite variable.
Yeah, and I suspect that it is something like that. I think there's no question that it has to be something like that. This is the kind of stuff that's now being done by groups in Denmark. And there's Uda Keen and Roger's work, who's at Cornell. They're working with genetic markers that they
how is it put together, how is it and so on. I think the problem is going to be that it's just very complex. It's just not going to be the kind of thing that, Western's worked on this gastric ganglion. And this gastric ganglion of the lobster is just not a spinal cord. It's going to be much more complicated. And, I think that's what has to be done. It's going to be a long time coming.
I would suspect that even the swimming dinosaurs might have a CPG of a very primitive sort.
Oh, it does. There's no question it does. The problem is that you can't take it out of the animal. And you can do that in a leech, for example, and it has been done. Not for a swimming leech, but the heart rate of the leech. Ron Calories does that in the Emery. There's all kinds of CPGs all over the animal kingdom, and many of them have been studied in great detail. One which is studied in much more detail than the motor CPG and cellular aspects is the respiratory system of us, mammals. And that's a beautiful system which is smaller than the double part of the brain. And it's a much less complicated system in some ways, but in fact, when you get into us, what happens is that the control of the, as you know, if you ever watched Christopher Reeve speak at his back, the breathing, when you speak, you are pushing air through the larynx, and the point is that is all part of breathing. But when after you broke your spinal cord, what happens is that and that was how we talk. Now that's not what we do. We have a much more complicated way of controlling our talk. So it's, in some ways it's a very simple system, in other ways it's extraordinary. So, and all of these systems have their pros and their cons. There just aren't any simple ones. I think what I would like to suggest in this talk is that what we have shown here is the kind of spread of expertise which is required for any one of these systems. Locomotion in us, well, there's Bob Full, for example, who does some beautiful work also with on the cockroach running. But even the system of the neural network for the cockroach running system it was done decades ago, and it hasn't been updated. I'm sure that it's much more complex than they thought it was. But, and Philip has done some beautiful work on that, because the animal, it can run over, there you get the complexity in terms of what the animal does is it essentially almost flies over things. It doesn't even bother with feedback. You've ever watched a cockroach run across something, it's extraordinary. As it moves the way it does for nothing. So the point is that all of these systems, every system has its pros and its cons. I agree with you that there are certainly better systems to work on. The advantage that we have had is, or that we claim to have, is that we're working on a vertebrate. It's the only vertebrate where you can put together the entire system and really look at it from the neurons all the way up to the vortices. And that's simply it can't be done with any other system. But even in that simple system, I couldn't do this way. Oh my God. I can't remember Reynolds numbers from the small number. It's just, fortunately I don't have to. But, it's impossible. I thought I was doing great when I learned the mathematics before. But, getting into the fluid dynamics, give me a break. There's just no way. So, and that's what's going to be required in any of these systems. So I think what the power of the systems approach is to put together a group of, across disciplines, working together in a way which can do this. And allowing the groups, to have this kind of power. It's only the way. So, yes. neurons.
That's right. Yeah.
So maybe somehow,
Yeah, there's no question that we have ignored this part of the model, the differences in muscle types. The Lamprey does not have the complex muscle types that mammals have. They have just slow and fast. And the anatomy of it is also simplified for us. But we've ignored that for now.
Not entirely. I mean, so we can alter the twitch dynamics of our muscle, and we get very different behavior if we slow it down or speed it up. Particularly if we slow the muscle down, so it takes longer to reach its peak tension relative to the cycle time. That really does change the behavior dramatically. So we're sort of getting there with that.
In simulation, you could do that. I think was a question about the actual animal. So in simulation, you can tweak this twitch time, but in the animal itself, that might not be possible, which is what I think the original question.
Well, we do have some recordings from honest-to-God animals which we can see what the twitch dynamics are. And Velma actually has done a little bit of that, and I've done a little bit of reduced vibration stuff, shall we say. And we also do have some intact animal recordings. We may have to go, actually, that'd be interesting to add to our next stage, is to do some real honest-to-God recordings in intact animals to get some of those twitch dynamics, because we should be able to make it in the next iteration. Thank you. That's a good thing.
Speaking of that, is there a preparation some more like the cat, where you have the animal without a brain that was most muscles to work?
Well, the neonatal rat is such a part.
I know that preparation
The
is
Lamprey,
involved
yeah,
with Lamprey.
we do actually. We have both cut off the brain. In fact, all of this is spinal cord, it's just the spinal cord for the brain. We also have a prep that we introduced in the 80s, which is what I call the body prep. And what you do is you cut off the head, and
Yeah, it's sort of George Bush Lamprey.
that's right. I didn't say that, Bill said that. But I'm happy to. Yeah. We won't publish that. So the first thing we do is we cut off the brain, throw it away. We also then have to peel off the skin, because the skin actually has toxins in it. So we peel off the skin, and we open up the spinal cord so that the spinal cord can get activated by a cocktail of drugs in the bath. And then there's this little body prep will swing, and then, and now you can play games with it. You can cut it so that it gives you two different frequencies. You can do,
Even closer to that prep, there's also a cerebral prep.
That's right.
Where you like higher brain centers, brain stem. You have to stimulate in the brain stem.
Can you make that guy swim?
Yeah, you have to stimulate electrically the brain stem to make it work, but yes.
Well, actually, Tim and I did it without. We had the brain stem. We used, it turned out to be a mess, because what you're doing is you're activating the spinal cord chemically, and the brain is seeing that and saying, what is going on here? So the interaction between the two is very abnormal. So it's not clear exactly. I think that the better way to understand it.
How big it is?
The big guys,
Okay.
yeah.
So the small ones are
The ones are tiny. The babies. The baby, the amicetes get up to about this, skinny. And then the adults start that size, and they grow up to about this.
So I've seen people finish it over like the head, and then image higher resolution images of body.
Yes.
That's a possible here with your system.
Yeah.
And then you need them to move through the needle, you just monitor.
Well, Feldman did that. Address that.
You can do free swimming experiments
Tether.
You can tether them. Apparently, most technique for doing that was actually to let it suck onto her thumb and stick the thumb in the fluid and let it swim.
Very sophisticated.
But basically, I mean, they're just, animal behavior becomes an issue, and they're sort of difficult to yeah, you can get much more repeatable behavior out of eels. The eel nervous system is much higher up the vertebrate and a lot more complex than lampreys.
Well, like I said, something to do with the fish, so I think that and
You're thinking of some more or
yeah, can you do
yeah.
that with
For these purposes, we haven't tried.
but you're aware of it. So.
Yeah, the convenience of doing larval zebrafish head in bed stuff is you still get some tail movement, but the animal is also transparent, so you can also image neural activity through it. Since we're not transparent, we lose some of those benefits. You've got to try.
Well, I mean, it's already for both of them. We're looking for actually.
Yeah, and there is, Joe Fetcher has done some stuff with the muscles and all the neurons of the lamprey. That would be huge. So it would be possible in him situation.
I was wondering if there were parameters you could change, for example, externally, the viscosity of the fluid in which the critter is swimming, or internally, using some sort of drug, the neuromuscular response figure, might be able to show that the same nervous system can result in effective swimming even under different conditions.
So we certainly can change viscosity. We've done some preliminary work there. And in fact, we do see some of the same effects that we get in simulation. So when the animal swims in high viscosity fluid, it does seem to go over body weight. Although I don't have data to really say that conclusively, but that seems to be what's going on. Changing internal parameters, I'm not sure how I go about that.
Alcohol. Relax.
One of the things, I don't know, I mean, I know calcium used as, in the cell exchange, uses calcium as, calcium channel and in the exchange. So, what is required to be there? So I wondered if something
The problem is that we just don't know how to determine calcium.
it does.
We're trying to keep it as,
Maybe you need calcium something else.
I'm sure you do, but that's the problem.
Potassium, you know, some kind of.
I would be very reluctant to try that because I think that it would be, you wouldn't as it is, we have a lot of unconstrained parameters, but that one would just be awful.
Yeah.
So, as it is, the ones we've got are not as often well as that. Yeah.
Generally, gender-related parameters that affect the dynamics of
Maybe what?
gender-related.
Gender-related? No.
Any what?
Gender-related.
Gender-related.
Now, the males and the female, the only thing that there is a difference is that the female, when she's getting ready to spawn, is filled with eggs and is really stiff. That, we never thought of that. But,
I mean, when I do my dissections, I always note whether they're male or female when I do the dissection, but I've never obviously noticed any systematic differences, and I've never gone back through the data to see if something falls
but it
out.
might be interesting to look at animals that are, yeah, right. Because, at that point, they really are, when you see the females,
There's some sort of persistence of behaviors on lots of critters. They'll find a different way of doing something if they're somewhat physiologically changed, handicapped, or
Oh,
whatnot.
absolutely, right. Well, that's actually, I tell my classes that, as we gain weight, look at poor Megan, she's been
Still able, she was still able.
oh yeah, she was doing fine, She was working out a week before having the baby. So, I was vertically challenged, but when it gets really tall, you start bumping into things until you learn where your head stops, and the parameters of running are going to change. So we know the spinal cord is capable of all these kinds of changes.
So does the model account for those changes?
Our models.
Well, we don't know yet. That's sort of the next step we're going to take. Suppose we keep the neural model identical. Well, I mean, I showed you some results there. We keep the neural model identical, change the body properties, we get different behaviors to some extent. We can start to look at the robustness of the whole system to these sorts of behaviors.
Yeah, and that's what we're planning to do in the next iteration of the grant. And as I was always asking for all of this interdisciplinary work. I mean, you can't get more interdisciplinary stuff. They like it once. I'm hoping that they'll like it way out. It really is, I think, it's a real power systems and interdisciplinary. So thank you all.
Well, thank you. Let's thank them.